Good afternoon, children of class 11th. Today in Bio, we continue with the study of chapter 8, which is Cell, the Unit of Life. And in video 1, we studied about cell tissue, about organ, oxygen system, and we see the cytology, cell biology, the history of contribution to the study of cell. We studied about so many scientists who contributed to the detailed study of the cell, and we also studied about the unique contributions, and we also saw the years in which the various historical advancements in the study of cell took place. Today we will study about the deeper details of cellular life. The cell should be a self-perpetuating chemical. In order to maintain the concentration of chemicals, the raw materials are acquired from the environment. So we see that the raw materials, they are acquired from the environment. There are membranes that are covering, or there are certain cell walls that are covering the cell, and this is the outside environment, outside the cell, children. And we see that the functions of the cell are accomplished by specialized structures which are known as organelles, like rooms of house permit specialization. Likewise, there is a division of labor in the cell among the organelles of the cell. We see the form assumed by organelle is related to its function. The structure and the particular shape that each organelle takes inside the cell, that is related to a function. We see that there are compartments in the cell, in the cytoplasm of the cells of plants, animals, and we see in protists and fungi, they have many membrane-bound compartments called membrane-bound cell organelles. The cell also has some membranous cell organelles without a limiting membrane. So there are both kinds of cell organelles. Those cell organelles which are bound by a limiting membrane, and those cell organelles which are non-membranous and do not contain a limiting cell membrane. Hence, two types of cell organelles are found in cell. As follows: membrane-bound cell organelles, these are like mitochondria, chloroplast, nucleus, lysosomes, vacuoles, Golgi bodies, and endoplasmic reticulum. Whereas non-membranous cell organelles, examples are centrioles, ribosomes, cilia, flagella, etc. We see that prokaryotic and eukaryotic cells are the two basic types of cells that are there. In prokaryotic cells, we see in Greek, pro means primitive and karyon means nucleus. So we see in prokaryotic cells the characteristic, this is the characteristic of bacteria, this is the characteristic of cyanobacteria, in mycoplasma, in PPLOs or pleuropneumonia-like organisms. It's simple in morphology. And we see that prokaryotic cells have a cell wall surrounding the cell membrane, except in mycoplasmas, which we have already studied before. Then we see that the cytoplasm, it lacks membrane-bound cell organelles in prokaryotes, and the respiratory or photosynthetic enzymes are located in the plasma membrane. There is no well-developed nucleus the genetic material DNA is not organized into distinct chromosomes but distributed throughout the cell. The cell membrane has higher selective permeability. A specialized differentiated form of all membrane is called the myonemes. It is the characteristics of prokaryotes. We see that myonemes is the characteristic of the prokaryotes. And then we see that the structure of bacterial cell shows typical prokaryotic structure. We saw the cell envelope just now when I was showing you the bacterial cell. The bacterial cell have a chemically complex cell envelope. The cell envelope, it consists of a tightly bound three-layered structure, as I showed you. The outermost is the glycocalyx. The outermost membrane is the glycocalyx, and then the middle one is the cell membrane, and the third one is the cytoplasmic membrane or the plasma membrane. And we see that in the cell membrane or the cell wall, the bacterial wall is surrounded by a tough rigid cell wall. It determines the shape of the cell, as you saw here, that this cell wall, thick cell wall, it determines the shape of the cell here. And we see that it not only makes a rigid cell wall, it also determines the strong structural support that it gives to the cell, so that the bacteria, they don't burst or they don't get crushed. Then we see the characteristic cell wall material of eubacteria is peptidoglycan. It consists of two amino acids which are N-acetyl glucamine or AGA, and the other one is N-acetyl muramic acid or AMA, cross-linked with short amino acid chains. Then we see in bacterial groups, they fall into two natural categories. The bacterial groups, they fall under two natural categories, as we see the difference here. We see that they are the gram-positive bacteria and the gram-negative bacteria. Children, here we see in this that Hans Christian Gram in 1884, he said that those bacteria that were stained with blue or purple color, these were the gram-positive bacteria, whereas those bacteria which did not retain the stain but they became colorless, these were called by Gram as the gram-negative bacteria. We see that the cell wall is 20 to 80 nm and is single layered in the case of gram-positive bacteria, whereas in gram-negative bacteria we see that the cell wall is 8 to 12 nm and is two layered. Then we see in gram-positive bacteria the mesosomes are more prominent, whereas in gram-negative bacteria the mesosomes are less prominent. Another difference, children, which we see here is that the pathogenic forms of bacteria are fewer, which come under gram-positive bacteria. But we see that the pathogenic forms are very high when we talk about gram-negative bacteria. Then we see that the in gram-positive bacteria, the lipid content of the cell wall is very low, whereas in gram-negative bacteria we see that the lipid content of the cell wall is 20 to 30%. Another difference is in gram-positive bacteria we see that the cell wall straight and contains teichoic acid, Whereas in gram-negative bacteria, we see the cell wall is wavy, it is not straight, and they do not contain teichoic acid. Then we see in gram-positive bacteria, they lack fimbriae or pili, and the example of this is streptococcus. Whereas in gram-negative bacteria, fimbriae or pili are present, example in Vibrio. Then we see another aspect of the prokaryotic structure is glycocalyx, or this the glycocalyx is either a capsule or it's formed of slime layers. And we see that the glycocalyx, it differs in composition and thickness among different bacteria. We see it is a gelatinous layer present on the outer surface of the cell wall of some bacteria, and it is usually composed of polysaccharides. This layer is usually composed of polysaccharides or polypeptide chains of amino acids. Then we see that this layer, glycocalyx, this layer, when composed of only polysaccharides, that time this layer of glycocalyx is called slime layer. Whereas when amino acids or polypeptides are also present along with polysaccharides, then this layer, glycocalyx, is known as the capsule. Then we come to another part of the bacterium, or another part of prokaryotes, which is flagella and pili. We see flagella are extremely thin, hair-like appendages that protrude through the cell, and these are responsible for motility of the cell wall, or the movement is produced by the flagella. And we see that these are responsible for motility or movement of the bacteria cells also. These are made up of identical spherical subunits of protein called flagellin. What are the flagella made of? They are made up of a protein called flagellin. The flagellum is rigid, and though it is shaped into a wave, it has a unique mechanism of action. The base rotates on ring-shaped bearings. We see that the base rotates on ring-shaped bearings in the flagella. Let us look quickly at another diagram which we have here. Very beautiful and clear diagram is there about the flagellum. Can you see here, children, the flagellum here? We see this is the basal body, and we see from here it makes a rotatory motion. The flagellum makes a rotatory motion. So we see that the base rotates on ring-shaped bearings, so that the flagellum does not beat, but it performs a corkscrew motion to propel the cell along. So it rotates, and the base of the flagella, it produces a corkscrew kind of motion to propel the cell along. Then we see what is pili. If we see here pili, these are mainly in the gram-negative bacteria. They possess minute rigid cylindrical appendages extending outwards from the cell wall. These appendages, called pili or fimbriae, are made up of special type of proteins known as pillins, and these have no function in motility. The pili, they have no function in motility, children, but the pili, they are concerned with cell or surface attachment, either cell to cell attachment or cell to surface attachment. It is with that the pili is concerned with, children. Then we see about the sex pili. In addition to the normal pili, children, there are some special type of pili known as sex pili. These are helpful in transfer of genetic material during bacterial conjugation. Then we see the next part is the cytoplasmic membrane. And this cytoplasmic membrane, it lies immediately beneath the cell wall. And this is a very, very thin membrane. We will continue about the details of the cytoplasmic membrane and the cytoplasmic inclusions in the next video. So till then, wish you all the best. Have a good day. Thank you.